Uh, okay, hello everyone. Thanks for joining us today. We have another webinar for you. Before starting, these are our upcoming events. You can join the Journal Club, or we have webinars from other professors, and we also have upcoming student seminars that you can also present if you want to. Please check out our website or social media links that I put on the sticky note on the right side. If you have any questions, we will take them after the presentation is done. Just type them in the chat box, and if you raise your hand, it will be easier to see. Uh, and for our speaker today, we have Professor Niranjan Nagarajan. Uh, Dr. Nagarajan is the Associate Director and Senior Group Leader in the Genome Institute of Singapore and Associate Professor in the Department of Medicine and Department of Computer Science at the National University of Singapore. His research focuses on developing cutting-edge genome analytic tools and using them to study the role of microbial communities in human health. His team conducts research at the interface of genetics, computer science, and microbiology using a systems biology approach to understand host microbiome pathogen interactions in the various disease conditions. Dr. Nagarajan received a BA in Computer Science and Mathematics from Ohio Wesleyan University in 2000 and a PhD in Computer Science from Cornell University in 2006. He did his postdoctoral work in the Center for Bioinformatics and Computational Biology at the University of Maryland, working on problems in the genome assembly and metagenomics. And without further ado, I will leave the stage to you, Professor Niranjan.
Thank you. Thank you so much for the kind introduction. Allow me a few minutes to set this up. Let's see, start screen sharing. Okay, I hope you're seeing my entire screen, including my own Firefox window. And then, okay, I hope you see the first slide of the presentation at this point.
Fine.
Yeah?
Yeah, everything's fine.
Okay, excellent. Thank you. All right. So thank you for this opportunity. It's really a pleasure to be speaking to you all. I visited Turkey only once before in my life. It was really a memorable trip, and I hope I can do so in the future. I'm based at the Genome Institute of Singapore, which is a part of Agency for Science, Technology and Research, ASTA, which is the leading national agency for research in Singapore. And we have a lot of trainees as well as researchers from Turkey. So it's a very vibrant community here. If you are planning to visit, please do let me know. We would love to welcome you here. So today I'm going to tell you about a bit about a topic which is perhaps gaining in prominence, but not the usual human genomics focused computation biology. I'm going to talk about microbiomes. So a quick primer. Microbiomes are really referring to the microbial biospheres that we inhabit. We live in a microbial world. We are only a small fraction of the overall biodiversity that's out there. And this genetic diversity really is there in the microbial world because they evolved much before us. So if you look at the tree of life, which is the figure shown on the right here, allow me a moment, I'll get a pointer, which is shown on the right here. What you should be able to see is that the eukaryotic lineage, which is seen in the bottom, is really a tiny fraction of the overall diversity of organisms out there. So microorganisms, which we cannot see with the naked eye, they are really a majority of diversity. In order to study this diversity, modern genomic technologies, in particular the ability to take total DNA extracted from a sample and sequence it, is really a powerful approach to get a snapshot view of the community, in the sense that you get for computer science terms, a vector where each entry of the vector is the abundance of a particular microorganism in the sample. So it's a bit like gene expression data, except that instead of genes, you have specific organisms that you identified in the sample. And based on this capability to get a whole community profile, we're increasingly trying to understand how this microbial diversity plays a role in human health. And one particular low hanging fruit that my lab is interested in is studying the role of this microbial mediators in infection. So when there's a pathogen, there's a specific microorganism that's playing a role in a disease, whether it's in the gut, in skin, or in cancer. How does the microbiome serve as a mediator in these host pathogen interactions that we traditionally just saw it as the view of invader species and the hosts defending against it? Well, in fact, really the microbiome, the whole community of commensals that inhabits our body, plays also a role in defending us. So as you can imagine, in order to be able to study something like this, we really need better tools and technologies, because what we are trying to do is differentiate between friend and foe, and even different strains of E. coli could have different pathogenic properties. So we really need improved resolution in terms of analysis. Nanopore sequencing, for example, long-range sequencing technologies have been a powerful new addition to our arsenal. But I won't be talking about this aspect. We're talking about something that's it's something that I see as the next wave of being able to understand microbiomes, which is really the ability to model microbial community dynamics. Because unless we can model how a community evolves and how a community responds to perturbations, we really can't figure out how to rationally intervene, how to rationally trigger, let's say, decolonization of a pathogen from the microbiome. So this is the next stage where, as our observational technologies improve, our modeling technologies have to keep up. And more broadly, the kind of medical problem that we are that our research revolves around is really the challenge of antimicrobial resistance. The fact that many of the bugs that we see causing infections are multi-drug resistant, multi-antibiotic resistant, and this issue is a whole cycle starting from hospitals where many of the severely ill individuals are treated, often with broad spectrum antibiotics, to the impact on the commensal microbiome as a function of this antibiotic usage, how this might predispose certain individuals to colonization by other pathogens. Which is ironic that the use of antibiotics itself might really lead to the emergence of multi-drug resistance as well as colonization of our microbiomes, and then on to an overt infection in terms of liver abscesses or other body sites. So we are trying to understand all stages of this cycle and the transmission patterns. And some of the studies we are doing include studying asymptomatic colonization. So this is a situation where there's a multi-drug resistant pathogen which can potentially lead to an infection. So these carbapenem resistant enterobacteriaceae like E. coli and Klebsiella, and these are known to colonize the gut and persist for extended periods of time, serving as the basis for onward transmission of resistance determinants like plasmids in mobile elements, or even onward transmission to other individuals. So we have a study where we're trying to understand these dynamics. And one of the things that was striking for us when we did the study, here what I'm showing you is the abundance of Enterobacteriaceae. So basically these vectors that I described of abundance of various species now converted into a stacked bar chart, where each stacked bar chart is one time point. So the numbers here on the x-axis represent different individuals, and each stacked bar chart, it is a different time point. So up to 12 time points up to 10 time points, per individual. So you can see as you go across time points, for example, for individual 4, 5, 7, the community of Enterobacteriaceae species in particular changes dramatically from Proteus species to E. coli and Klebsiella towards the last time point. And similarly for other species, for example, 511, between 2 time points is a complete shift from having E. coli dominant gut microbiome. And this is quite unusual because E. coli is not one of the more abundant species typically in the gut microbiome, all the way back to a Klebsiella pneumoniae dominated gut microbiome. So this sort of dynamics is really intriguing, and we hope that understanding these dynamics can help us actually boot out these multi-drug resistant bacteria from the gut microbiome. Another study that really kind of drove home the point for us that we need to really understand how microbes interact each other, really the ecological dynamics of the gut microbiome, a complex population of hundreds, if not thousands of species, is this phenomena of gut microbiome recovery. So when you take antibiotics, in addition to treating the infection, there is collateral damage, and that is in the form of the gut microbiome, whose diversity goes down. By diversity, I mean the richness of species and their proportions in the community. It suffers substantially. On the y-axis here is really the diversity, and on the x-axis is time across individuals. And you can see for many individuals, there's a U-shaped recovery. So the diversity goes down when you're being treated with antibiotics, and when the antibiotic usage is stopped, the community recovers to what you expect to be in the original healthy gut microbiome state. But there is a subset of individuals where this does not happen. The community diversity drops dramatically, and then it stays low for extended periods of time, up to a year in some individuals. So we wanted to understand what differentiates recoverers and non-recoverers. And of course, there could be many factors that differentiate the two, but we started looking at microbial factors, and we found that there are quite a few microbial factors that consistently show up across cohorts. So from 4 different cohorts with a whole different set of antibiotics and environmental factors playing a role, we consistently got the same signature of certain species playing a critical role in being associated with the recovery of the community. So this was quite interesting as an initial insight. And so we try to understand this further in a causal model. So we had a mouse model where we gave the mice antibiotics for several days, and then gavaged them with specific probiotic species that we identified. So Bacteroides theriadomicron as well as Bifidobacterium adolescentis are these recovery-associated bacteria that we did with this association analysis. And we also tried a combination to see if the synergy works and compared them with just a PBS vehicle control. And what was striking really for us is that mouse studies can be amazingly finicky to work with. But in this case, if you looked at the bacterial biomass, by day 6, the antibiotics are stopped and we started to give the probiotics. By day 10, we see a dramatic increase in recovery, improvement in recovery in the two cohorts which have Bacteroides theriadomicron, and as well as in another cohort, another set which has Bifidobacterium adolescentis as well. So there's a dramatic microbiome recovery, which is not triggered by just the probiotic. For example, just Bifidobacterium adolescentis, a classic probiotic that's there in some of the probiotic tablets that you take. And BT on its own also, the vehicle on its own also does not lead to recovery. But in terms of another aspect, which is the diversity of the community, as I said, how rich and how even the community is in terms of its profile, that's a marker of a healthy gut microbiome. And again, we saw that diversity is triggered by some of these combinations, more specifically the BT plus BA combination, the synergistic combination, seems to improve recovery much better, especially by day 22. So this is a tantalizing insight for us that there is some interaction which is important. And so based on this and based on a lot of other analysis that I'm not showing here and reported in this paper, we believe there's the following model that plays a role in microbial recovery after antibiotics. There are certain species which we call primary RABs, or recovery-associated bacteria, which are have the capability to degrade host-derived carbohydrates, so the mucins that the host produces, as well as complex dietary carbohydrates, and breaks them down into simpler carbohydrates that other bacteria can use for their energy. And some of these other bacteria, which we call secondary species or tertiary RABs, they can produce specific molecules called short-chain fatty acids, and these short-chain fatty acids serve as energy sources for colonocytes in our gut. And of course, as the colonocytes differentiate and they ramp up their there's more colonocytes in the gut, there's more mucin that's produced. So this is the basis of a positive feedback cycle, which really ramps up the recovery process. So there's this cross-feeding bacterial food web that's important to understand. And while we can imagine using pills, specifically maybe for Bacteroides theriadomicron, which is obviously an obligate anaerobe and therefore hard to construct a pill for, we could also imagine feeding our the specific bacteria that are important. And there's quite a lot of study, though still a bit of controversy on how diet alters the microbiome. And there's several hundred studies now since the 1970s which have studied this topic. But yet, all of this is a low throughput exploration of this vast space of how the specific components of a diet, with their diverse metabolites that they can provide to our gut bacteria, how that promotes different constituents of the gut microbiome, how they can shape the gut microbiome. And so, this is really a systems biology problem in our minds. And we think this is one where a data-driven approach, where not just specific targeted experiments in small cohorts or in vitro studies in reactor models, these alone cannot answer the questions that we have. So this really motivated us for some of the methodological work that I'll describe next. In particular, I'm going to try to address two major problems for microbiome modeling. The first is the compositionality of the community, and I'll tell you a bit more what that means. And we had a few methods for that. One approach that we call BEEM that really works well. The other is really the availability of data. No modeling method can work well if there's not enough data, and so we need to relax some of our assumptions in being able to get enough data for modeling work. Okay, so a quick primer again into microbial interactions. Microbes interact in different ways. Some inhibit others, and we indicate that with a plus sign. Some minus sign. Some support the growth of others, and that's indicated with a plus sign. And the zero indicates there's no effect. And of course, does not have to be symmetric. One bacteria can promote the growth of another, while another can inhibit the growth of the first one. So that's shown in this predation model, where it's a plus minus interaction. And so there are 6 different classes of interactions that have been described at least in a binary sense. And these can be also viewed in terms of metabolic interactions, so either a chain or a competition in certain way. These different microbial interactions can be mediated by a food chain that we'd like to reverse engineer from the data. So that's really the problem that we want to set ourselves up to, because this is the foundation for a lot of a systems understanding of what happens in the context of the human microbiome. And classically, this sort of ecological interactions have been studied by a very simple model, a predator-prey model. So you might have seen this if you have taken an ecology class. But this is just intuitive, right? Let's say there's a predator, which is, in this case, a fox in this graph, and a prey, which is a rabbit. As the abundance of rabbits goes up, of course, there's more food for the foxes to eat. So the fox population grows up in this blue line. But as enough foxes are there, they start hunting the rabbits really well. The rabbit population starts collapsing, and in tandem, the fox population will also collapse. So this process can cycle, and there are equations to describe it, but of course, microbial interactions are much more complex in the sense that there are many species that are playing a role, and we can't directly observe them. So the modeling approach that we're going to rely on is something called generalized Lotka-Volterra. So it's generalization of this predator-prey model. First order differential equations, where we model the rate of growth of species I, dxi dt, as a function of the abundance of species I, the absolute abundance, multiplied by a constant term mu I. This models sigmoidal growth. And in addition, a whole bunch of interaction terms over all other species, j equals 1 to p, of beta ij, which is the interaction term, xit, which is obviously the abundance of species I, but also the abundance of species j, which might be influencing the abundance of species I. So this is a very simple linear relationship. To a first approximation, quite useful. Of course, also pairwise interactions are only modeled here. Now, so this model is a classical model, and it's been applied in small scale experiments. But as we start thinking about metagenomics and how we can use metagenomic information for microbiome modeling, we have to realize that there's one piece of information that we're missing, which is that metagenomic count data is is a relative abundance. It's not an absolute abundance. And that means that we're missing a critical piece of information. So when you have these two taxonomic profiles, one where there's an orange and a green species roughly equally proportional, and in another profile, there's more orange species as relative abundances, and this is what you get based on count data that you generate in sequencing. We don't know really what happened in terms of the absolute abundances. It could have happened that green species went down in abundance, or the orange species could have gone up in abundance. And so, and of course, both of these could have happened. We just don't know. And this is not just a problem of microbiome data, it's a problem of any sequencing data. So transcriptomics data has the same problem, except there is a smaller problem there. There are many more genes in a transcriptomic profile. In a metagenomic count profile, you might have a small number of species, let's say 50 or 100, and some of the species might be a lot more abundant than others. And so this skew is a bigger problem there. So you would imagine, well, okay, we are off by a factor, right? And this is a factor, a scaling factor, really, right? That gets you from relative abundance to an absolute scale. Why not just measure that directly, experimentally? You can count the number of cells that you see in a particular sample, but you can imagine the sort of counting is experimentally challenging depending on the matrix, the sample you're handling. Of course, if you're handling stool samples for studying the gut microbiome, this may not be a very amenable assay. There's a more traditional molecular assay, which is the 16S ribosomal RNA. It's a universal marker for bacteria especially, and you can amplify it and you can quantitate it. But there is quite a lot of experimental error, and this is data from a published study. When we look at the replicates that were used, the technical replicates, the correlations can be quite poor. And you might say, well, okay, so what? Maybe you can still learn the model quite accurately. And so we generated some synthetic data sets which mimicked some of this, where the data is really from the generalized Lotka-Volterra model, and we knew the true biomass that we simulated with. So, and we used that to relearn the parameters. So with that, of course, you can get pretty good accuracy of parameters. So growth rate is the alpha i's that I mentioned earlier, and interaction parameters are these beta ij's. Interaction network structure is just converting that to a 0, 1, whether a beta ij is non-zero or it's 0. So in this, all these three assessments of how well our ability to infer models from data is, if you know the true biomass, and in the simulation we do know the true biomass, you can get pretty good estimates of all these parameters. The interaction network structure, you get it with AUC of 0.9, so that's really darn good. But if you use qPCR data with these noise characteristics to estimate the true biomass, then the model that you learn is really way off. A relative error is worse than, in fact, random, which is shown by this gray line here. So you really get no information with the error that's there in qPCR data. It does not matter if you had one replicate or you had three replicates. So rep3 indicates that you had three replicates that you average to try to get a more accurate measurement. And so this is a consistent problem. So, well, what does this mean? Does this mean that we really cannot do this sort of modeling? Of course, you would not be in this talk if that was true. And we try to do something crazy, which is that, can we learn these scaling factors that we don't know along with the parameters of the model? So we're now increasing the number of things you want to learn, and you just hope that the data is sufficiently constraining of the model that we can learn these parameters. So a reminder, so dxi dt is the rate of change of abundance of species I. You can estimate this from time series data. If you have 2 time points, you can sort of you can differentiate the data numerically and get dxi dt. Mu I is the constant that you want to infer, which is the kind of the sigmoidal term, and beta ij is the interaction term that you want to estimate. Okay, so let's do some math jugglery. First, we'll move the xit from this equation, so we get it to the right, the left-hand side. And when you have 1 over xit, it's just the derivative of the natural log of xit. So this is just calculus, and now we don't have xit on the right-hand side. So that's, at least we converted something which has quadratic terms with two xi and xj. We just left it to have just xj on the right-hand side. Um, Then, the xj is in reality for Lotka-Volterra, it's the absolute abundance. So you want to convert it back down to relative abundance. So x tilde I is the relative abundance that you can measure. But of course, x tilde I needs to be multiplied by the scaling factor, which is a sample specific, time point specific scaling factor, mt, which is the absolute biomass in my mind. So mt times xi tilde gives you xi, right? So it gives you the absolute abundance. So of course, this when you have a log of a product, you get a log of sum, so you get this nice term here. And on the right hand side, you can just take the m of t term out from all of these, from this whole summation. So this is what you get. Then we do a neat transformation. We basically do a log ratio transformation. So basically divide the whole equation, the previous equation, with the same equation for another reference species, x tilde r. So when you divide by the whole reference species, if you what you get is basically d dt of log xi tilde over log xr tilde. And so we are basically taking a log ratio transformation. What this does on the right hand side is that, well, on the left hand side, notice that now I no longer have the biomass term. So I eliminated the biomass term because subtraction of xi and xr removes the shared biomass term. On the right, I still have the biomass term, but now I'm purely in terms of these x tildes. I just reparameterized ai and bij, which are functions of mu I and beta ij. So I've rewritten equations such that the biomass term mt is only on the right hand side, and I have log ratios on the left hand side. I can still estimate these from the data. Okay, now what do you do when you have a situation where you have you have a set of parameters ai and bij, and you have another unknown data that you need to estimate? Well, those of you who have taken bioinformatics algorithms courses might have heard of expectation maximization. It's just a very neat trick which says, under certain conditions, if you set things up just right, you can iterate between learning the model parameters, which are AIT and BIT, with an assumption for the missing data, the MT from the previous iteration. And then once you have the model parameters AIT and BIT, you can go back and estimate new estimate of the unknown missing information, which is the MT here. So both of these have the form that they are a sort of regression, and they can be efficiently solved. And you can ping pong between the E step, the expectation step, and the maximization step. And under certain conditions, this converges rapidly. So this is the case for in this case. You need an initialization. Of course, you have to start somewhere. And you do that by initializing the biomass or scaling terms with just a very traditional metagenomic scaling approach, cumulative sum scaling. And that provides a very good starting point to do this EM algorithm. Okay, so let's, without further ado, let's jump into the results. And here's what you get with this approach. So repeating the same set of experiments with the same simulations that I described earlier. I'm going to first start by comparing these true results with what are we calling CSS, which is the initialization start point for our algorithm, the no biomass, which is our results. The CSS, or even a very simple total sum scaling, which is just working with the relative abundance as is. You can see that if you did that, the performance is quite poor. The relative error is quite high for both growth rate and interaction parameters, and the network structure is almost as close to random in terms of its performance. As before, the qPCR, even with 3 replicates, is not much better. And then finally, our approach BEEM, which does not require biomass estimates, can do as well as when you know the true biomass. So this is almost like magic. Even without experimental data, we can recover performance which is as good as having perfect experimental data. There is a little bit of a penalty, especially in terms of interaction network structure, because some of the rarer interactions or less strong interactions are harder to estimate accurately. So there's a numerical challenge here. But for the most part, in these simulations, Beam does really well. So we were very excited when we had this. Of course, when you do simulated um, benchmarks, you're just trying to get a sense that something can work, but really the proof of the pudding is when you start working with real data. So um, we leveraged some really nice human gut microbiome data generated from Eric Arm's lab at MIT, where they did daily sampling of the gut microbiome for 2 individuals. There's also a few other studies which have this property, so we exploited some of that data. And the this data was previously not really amenable to microbiome modeling, because even though we had daily sampling, we did not have a scaling factor. So as I'd shown you earlier, if you don't have the scaling factor, this biomass scaling, you can learn models. You can always learn models, but they are nonsense models. They're crap models. They don't tell you anything more than what a random model would tell you. Um, but with Beam, we have a chance. There's of course no guarantee that you have learned the right model, but we get estimates. We get estimates for biomass as well as model parameters, and we make some interesting observations about them. So one of them is that even with these 2 individuals, we see 2 different trajectories for biomass. And this sort of measurements of biomass, how many microbial cells are in a certain gram of stool over many days, almost a year, has not been done before. And we noticed some nice cyclical pattern in one of the individuals, almost seasonal pattern. This has not been described in Western populations, but it has been described in hunter-gatherer populations, particularly from Africa, where they do have dramatically different diets over time. But so it was interesting to see a similar pattern in a Western population. Um, of course, this is n of 1. But then once you look at the network structure that you got, so this is the network structure for the individual on the left in the previous figure, and this is the network structure you get for the individual on the right. Each node is a species. We just labeled the family name for the species. So I'm not giving you, sometimes I'm giving you the species name. So Bifidobacterium uniformis is a species, Faecalibacterium progenitori is a species. And edges between them indicate interactions. Solid edges are positive interactions, and dashed edges are negative interactions. The width of the edge also tells you the strength of the interaction, and the size of the node tells you the abundance of the species in the community. So the size of the nodes is log scale, just to be able to draw this without a very messy figure. But you can quickly see that in the case of the first individual DA, there are seems to be 2 hub nodes. These 2 hub nodes are your classical good bacteria in the gut microbiome, very keystone bacteria. So Faecalibacterium progenitori is a butyrate producer. Bacteroides uniformis is a Bacteroides species which has many functions in terms of carbohydrate degradation. They seem to be linked in one way, but it's a negative interaction, but they are not the most abundant species. There are other more abundant species in the gut microbiome. And similarly, for individual M3, there are more abundant species, but the hub nodes seem to be these 2 species, which are not as well studied, and they seem to have a positive feedback loop. So this is quite intriguing, given our observation in the previous graph of this very stochastic nature of the graph. Positive feedback loops are known to be unstable, and this could potentially explain the biomass variability that we observed for this individual. But taking forward this observation that the shared property that the core node of core nodes or hubs of these 2 networks, which are very different in structure, but they both tend to be abundant, not so abundant core nodes. We notice that if you look at the core species of the gut microbiome in a large public data set, those core species and the abundant species don't necessarily intersect. So they seem to be almost disjoint. This suggests a model of the gut microbiome we think is very plausible, which is that the core resident gut microbial species are the ones which are associated with the epithelium of the gut microbiome, and not the lumen, the spaces in between where maybe food and other particles and other microbes transmit. Those are more transient, while the core species are more stable, and they play a big role in determining the architecture of the gut microbiome. Okay, so I'll switch focus into the second part of this work. This is an extension and addressing another challenge that I think is quite important for microbiome modeling. So gut microbiomes are a kind of just a prototypical example here, just because they're so widely studied, especially in humans. If you look up NCBI, you find that there's nearly 5,000 bioprojects out there. But nearly all of this is cross-sectional studies. So if you just simply search for longitudinal studies with a few keywords, you get only a handful of projects. So it's a very small set of studies which have many time points. And these time series are also not very long necessarily. The ones I showed for that are year long are very rare to find. So how do you fit a dynamic model with cross-sectional data? Right? So if most of the data that you have is really cross-sectional, and your models only fit time series data where you can estimate dxi dt using numerical differentiation, then how useful are these models? And so we challenged ourselves and said, well, can we do something cute or interesting in terms of cross-sectional data? And yes, we can, but we have to make a very strong assumption. And the strong assumption at the starting point is that the model is at equilibrium for all the species, or many of the species that we are trying to study. So dxi dt is 0. You don't need multiple time points. And then you can simply write your equations like this, where the xi term, at least along with mu I, disappears, and the right hand side is 0. You can do a little bit more of math jugglery. I won't go into details, but the net result is that you get an equation very similar to BEEM, the original BEEM that we had, where you have relative abundance of species I as a function of relative abundance of other species, and there is a biomass term M. Long story short, you can use an expectation maximization approach. Here, I'm just going to give you a bit more details. These are nice linear regressions that you can set up. But of course, you can be a little bit more sophisticated. You can introduce a regularization term to ensure that you learn sparse models. So you try to push as many of these variables, these parameters, to 0 if possible. And so that's one approach to learn these parameters. And then when you have to estimate the biomass term, again, you can write a very nice straightforward equation for the biomass term. But this is actually for every species in the sample. So you get an MIN for every species I in sample N. And then you just have to take a robust estimator. And this is always the case when you have noise and the model does not perfectly capture the dynamics of the data that you have. So we have a robust solution which is based on the median. And as I suggested earlier, you can use this EM algorithm that goes back and forth between regression for the model parameters and just directly estimating the biomass term. And finally, you get the full model. Note that A and B are not exactly the mu and beta ij's that we had talked about earlier. They are slightly off. They're missing some scaling factors, but they are a good proxy for these original terms. Okay, so there was a huge assumption in what I started saying. What if that the microbiomes are at equilibrium? But what if the microbiome is not at equilibrium? What do you do in that situation? Well, here's perhaps a lazy solution, but something that could work. You identify samples that are not at equilibrium using a very stringent threshold, that the deviation of that sample from equilibrium is relative deviation is greater than 20%. If that's the case, remove the sample from your training and continue learning the model. So we can codify this into a bunch of equations here. What I'm showing here is xi tilde star is the estimate from the model of what your relative abundance should be given the species that are present in your sample. And you can compare that to the observed relative abundance, and the relative error, the relative deviation of that should not be greater than 20%. And that's the default. And this does a reasonable job of eliminating samples which are not at equilibrium. I'll show some results later. Um, but there's an even bigger challenge. I assumed that all data in your that you have in your database is coming from a current model, one Lotka-Volterra model. But what if the data does not come from one Lotka-Volterra model? You need some way to address that. And of course, as the data sets get bigger, this is more and more likely. So you can do a variation of what I described earlier. It's just that you have to open your aperture a bit and relax a bit. So you compare deviations with values predicted by model. So every data point has a deviation from the model. Some data points have small deviations. You think they're still coming from the model, but some have very large deviations. Their z-score of deviations is very high, greater than 3. This is a classical outlier detection problem. So we use that approach where we take the median of the deviations that we observe, subtract that, and divide by the interquartile range. And if that's greater than, let's say, 3, so like 3 sigma rule, then we say, well, this is an outlier. This is likely not from this model, and we remove it while learning the model. So this sort of bootstrapping approach of eliminating, learning an initial model, eliminating samples that may not be coming from the model, really works quite well. And this is again through simulations that we can assess this. Up to what I'm showing here on the left-hand side is the graph when the samples are not at equilibrium. And so up to 55%, as you can see on the x-axis, is not at equilibrium. And on the y-axis, I'm just showing you the reduction in performance from the ideal case performance. So BEEM-Static, which is our extension of BEEM, has a drop in performance, but with samples not at equilibrium, it doesn't go even beyond 5% in terms of drop in performance, while the naive approach can go up to 20% and even beyond in terms of a drop in performance. So we have substantial robustness when the sample is not at equilibrium. When the sample is not even from the model, which is what we simulated on the right here. Again, BEEM-Static shows substantial robustness, though at 55% of samples not being from the model, we start suffering, but it's still quite better than a naive approach where the error can go up to 60% reduction in performance. So we think this is still a useful approach even when some of your data is not from the model. And by learning the model and seeing how good it fits the data that you think is from the model, it gives you a sense of how useful the model would be. Now, the competitor for our approach is really a classical set of methods which are correlation based. So the idea here is that given these taxonomic profiles for two individuals, you can compute or for many individuals, you can compute the correlation between the abundance of species I and the abundance of species J, where if the two abundances go together, it means that they're correlated, and that correlation might be they are supporting each other's growth. So they actually have some sort of biological interaction, though it's a vague definition. And if they're anti-correlated, they're inversely correlated, maybe one is inhibiting another, but we don't know which way they are inhibiting. So these correlation-based methods have been widely used in metagenomics, for those of you who've been are familiar with the field. And there are various flavors of them. Some of them use correlation, some of them, like the Pearson and Spearman methods shown here, some of them use a covariance approach to account for the fact that the data is compositional, and some of them use an inverse covariance approach to account for the fact that some of the interactions may not be direct, they might be transitive. So A interacts with B and B interacts with C, but that might falsely let you conclude that A interacts with C. But as it turns out, if the data really comes from a generalized Lotka-Volterra model, then none of these approaches really work, as I can show in this figure based on simulations. Our approach, BEEM-Static, is again about as good as knowing the true biomass, so it eliminates the need for experimental measurement. While all other approaches, even though they are addressing interesting mathematical problems, if you believe your data is really ecologically driven and generated, not just correlation driven and generated, then these models do not do much better than random. So we this is quite surprising to us, and I think it's increasingly becoming apparent to the field that there are very strong assumptions in correlation-based methods. Of course, there are assumptions in model-based methods too, but at least some of these assumptions are seen in biological data sets, and they're known to be real kinds of interactions. So we unfortunately, there are not that many large data sets where we know the truth. And so we try to do as much as we can with existing data. So this is an example of a data set where we do know the truth, and it's real experimental data. We have some pairwise interactions, which are the ground truth on the figure on the left, and our method is on the has results on the right. And as you can see, our method largely recapitulates the direction of interaction and the strength of the interaction as well, except for a few missing interactions, a very weak interaction here that's missing in our figure. But in comparison to a correlation, in this case, based method, you can see a huge difference in the sense that we have bidirectional edges, and there's only undirected edges in this case, and there are some false positive edges. So in situations where, for example, there's two edges, where one is a promoting edge and another is an inhibitory edge, it really makes a big difference in biological interpretation compared to this edge, which just says, oh, there's a negative interaction or there's a positive interaction in the case between these two species, which does not really tell you why that comes about. In terms of further validation, we looked at some other aspects, how well our approach captures data types which are not from the model. So, for example, if you're taking antibiotics, that's clearly not, those microbiomes are clearly being perturbed. They're not at equilibrium. And so we have about 88% sensitivity to pick those samples up in real human gut microbiome data. We can estimate a biomass, as I had mentioned earlier. And we don't have a truth set which is super precise here, but we can at least see if adults have a higher biomass than newborns, and that indeed is the case. In fact, even the ratios in the right ballpark of what we know of adult and newborn gut microbiomes. We can also take all of this data, this 4,000 data sets that are uniformly processed that we had access to at that point of time, and try to learn models. And as it turns out, we do learn two different models. Our first model has about 2,000 data points, and the second model has another 1,500. And there's few more data points that that are left behind, but we can't model because there's just too few data to learn a 40 species microbiome model. And these two models are distinct in a few ways, but one of the ways they're distinct are the carrying capacity, the equilibrium biomass of two key species, so Prevotella copri as well as Bifidobacterium adolescentis. So this sort of distinct gut microbial equilibria in human gut microbiomes has been described before in the context of what's called enterotypes, but those have been derived from a clustering approach. So you would do something like a principal coordinates analysis to project this high dimensional data into 2 dimensions, and maybe observe certain density patterns, some dense clusters on the left and on the right here. But what we are showing here is that our models, the blue and the yellow models, they don't actually cluster in these PCA representations. They are actually capturing something else that's not captured by these original clusters, suggesting that there's some other biology that is there, something about ecological interactions that remains to be explored. So we think this can be another organizing principle when we start studying these sort of ecological models and how they are perturbed. So I hope I've given you a flavor of how we are trying to advance this field into practical applications. There's still many open directions. One of them is, what if we took the best of both worlds? So, the cross-sectional data is great. There's a lot of data, but it's really aggregating across many individuals. So you're learning some really averaged model, when in reality it might be that every individual has slightly different dynamics that's dictated by their environment, their diet, and so on. While longitudinal data is can give you individual specific dynamics, and also can really help estimate derivatives accurately, but it might be limited in data. So can we combine the strengths of these two data types? Can we extend these models really capture the fact that there are metabolic interactions which may not work in the classical predator may prey framework, and some of these interactions might also be higher order interactions. So there's some nice work in the space that we think can really uh, be complementary to what we have done. And finally, we think we're just starting to generate these large data sets that can be used for this sort of modeling. In our own lab, we have extensive work in the skin microbiome space. We're also looking at environmental microbiomes, particularly in hospital environments. We think it'll be very exciting to use these models to predict something that's useful, as a probiotic to improve skin health, or maybe as a competitor for pathogens in hospital environments, so that we can outcompete them and basically eliminate them from the environment. A direct one-on-one competition may not be sufficient, but together with the whole community, it might be a powerful effect in terms of outcompeting a pathogen. So, in summary, I would like to highlight that we think microbiome modeling is increasingly becoming feasible with metagenomics, even without biomass measurements. We think this is a big win for the field. And there's individual specific dynamics that we can capture. Cross-sectional data can also be used with its caveats. BEEM-Static as a method is robust to model violation. We have distinct ecological equilibria in human gut microbiomes, which are different from the enterotypes that we have heard of before. And the software that we have generated is all publicly available, and our data sets are also available. So please do reach out in case you're interested in using them, or you have any challenges in using them. So with that, I'd like to acknowledge Chen Hao, whose graduate work really led to all of this work, and you can perhaps get a sense of how much can be accomplished by really a motivated graduate student. We have open positions. If you or any of your friends are keen to come to Singapore, please get in touch. Would love to talk to you about it. My colleagues at GIS are clinical collaborators who have been kind enough to entrust us with their data, as well as the global community, MetaSub, which has been a wonderful interaction, CAMI, Oxford Nanopo, as well as the National University Hospital. Okay, so with that, thank you so much for your attention. I know this has been 50 minutes of a whirlwind tour in microbiome modeling. Hope you found that interesting, and I'm happy to address any questions. Thank you.
Uh, Thank you so much for that very insightful talk. It's really interesting that you can use cross-sectional data and model it after, I guess, similar to longitudinal data to
Mhm.
see how the gut biome is interacting with each other. Uh, I ha- I have a question, but if anyone in the audience has a question, please you can type it on the chat box on the right side. Uh, so for now, my question: is there a time limitation of how much we can gain information by using the cross-sectional data? Like, does it work for long-term data better, or for short-term data better?
Yeah, I think we haven't really fully investigated how much the models that are learned from cross-sectional data can predict time series evolution, so evolution over time. This is an open question. We suspect that it's largely good at only predicting at or near equilibrium. So how does the community evolve to small perturbations? We think in gut microbiomes, that might be often the case, that you're at or near equilibrium, especially at the time points of sampling. Um, but yeah, so I think what you're saying about how long term can you predict, even with longitudinal data, we think there are limitations with current models, and we think that's a function of the accuracy of the data that we have access to. That the relative abundance that we estimate has still a pretty large error bar on it, and so when we did some experiments where we sort of try to forward infer the community, I think after 3 or 4 or 5 days, the predictions are not as accurate as you would like them to be. So there's still work to be done in being able to predict community dynamics over time. But I think for us, right now, it's really very gross perturbations. Like, can you predict that the addition of one bacteria suddenly collapse the community in a certain way? So we're really looking for more dramatic changes rather than more subtle changes.
Okay, thank you. Uh, uh, do we have any questions from the chat? Uh, uh, I can ask another question. I'm
Sure.
not sure if this is too related, but I'm not very familiar with bacteria, but I mean, I know the viruses, for example, they all have different molecular clocks, so they usually evolve at different speeds. Is that affecting your model somehow? Let's say we there's a antibiotic treatment, and then some of the gut biome gets to back on its feet faster than the others. Do you have ways to show this in your models?
Yeah, so there is a, as I mentioned, the growth rate parameters there, mu I, which tells you how rapidly a particular species in the gut microbiome replicates, right? So
Mhm.
that parameter, yeah, does capture that aspect of how quickly something could repopulate. But of course, while it's repopulating, it's just like the fox and rabbit example that I gave you, right? While it's growing, its growth might actually promote somebody else, who in turn might kind of have a breaking effect on the first species, right? So these complex interactions
Mhm.
are typically what ensures that no one species outcompetes the others, right? Of course, there's what the media provides as food to the food we eat, how that promotes the growth of various bacteria. That also plays probably a role in keeping the community uh, uh, diverse and not having one bacteria outcompete others.
Okay, thank you. Um, I'm assuming we don't have any questions from the audience. In that case, thank you so much for coming today and giving us a very nice talk. It was very interesting, and thank you for your time. Thank you
Thank you.
for joining.
Pleasure, and thanks for your attention. Yeah. Yeah.
Okay. With that, our webinar is ending. Thank you everyone for joining. And if you're interested in more talks, please either go to our website or visit any of our social media accounts, and you can find information. Thank you again, and have a nice day.
Bye.
Goodbye.